Right, so in this lecture we are going to talk about incomplete and codominance. We've already spoken about Mendel's three laws of genetics, and now we're going to be talking about these other two ideas which are important when we are assessing the role of inheritance. So if you take two F1 hybrids, they might actually not look exactly the same as the phenotypes of the two parents. And this is quite an interesting effect. One of the classic examples is in snapdragons, which are plants, and they produce flowers. And in these plants, we see that we can have red flowers or white flowers. Red flowers are homozygous dominant, as we think they are, and white flowers are heterozygous recessive. But there's incomplete dominance here, and that has an effect on the offspring. So if we have our parents, one being the red flower, the other the white flower, we have our usual cross, which looks something like that. We lay it out in a Punnett square. But the result is slightly different, and this is because on the white flowers there's incomplete dominance. Right, so this is what it actually looks like. We have the red flowers on the left and the white flowers on the top. But because of this allele up here, what we see that all of the offspring, as we would expect, are heterozygous. But they're not heterozygous red, which is what we would think based on the laws of dominance, but they are all heterozygous pink. So essentially there has been mixing in the sense that the red has been diluted in a way. It's one way to think about this. But all of the offspring are heterozygous pink rather than heterozygous red. If we have a look at this in a visual format, this is what we get. So the parental generation is up at the top, and we can see we've got white and red flowers. We cross them, and all of the offspring will be heterozygous pink. Then, if we cross through to the F2 generation using the hybrids, so two of the heterozygous pink flowers, what we start to see is that we get a quarter, 25% of the individuals will be white flowers, will have white flowers. 50% will have pink flowers and 25% will have red flowers. But if we back cross with a heterozygous individual and a homozygous white recessive gene, we'll see that we get half and half. And that is because of the incomplete dominance of the white allele. Right, now the next important concept which we need to talk about is codominance. And codominance is when we have two alleles which are expressed in heterozygous individuals. So essentially they're being expressed at the same time. And a very good example of this is in our blood type, the blood type that we find in humans. So blood type A, which some of you may have, looks like this, either IA, all right, or IA and little I. So we have a homozygous A or heterozygous A, right? Okay, so that's how they can be written. Blood type B, very similar, so homozygous B or heterozygous B. And the third blood type, which you all should know about, is blood type AB, and that looks like this, right? You're starting to see how codominance works because we can have blood type AB. They are both dominant, okay? And that's why we have blood type A, blood type B, and blood type AB. But we also have another blood type, and it's blood type O, and that is the recessive blood type, denoted by II, little I. Okay, so we have codominance. So when we have the two dominant alleles being expressed together, we have type AB. And this makes for some very interesting problems to figure out for tests and exams, but also some interesting outcomes when we cross individuals with different blood types. So let's have a look at a codominance problem using blood type. If we have a homozygous male, type B blood, remember homozygous is BB, and a heterozygous female, right? So it will be with type A blood, AI, little I, we cross them. This is what it will look like, right? Sorry, here we go. We can have half of the individuals will have blood type AB, and the other half will have type B. No type O, which is very important because of the codominance effect, right? So we can have AB and type B because we're dealing with a homozygous male and a heterozygous female. So it would be half and half. Now, let's have a look at another codominance problem and see if you can figure out the answer. Here in our example, we have a male with blood type O. I'm actually blood type O myself, so it's small I, crossed with a female with blood type AB. What do you think we're going to get? Right, we set out our Punnett square and we lay it out like that. What are we going to get? Any O's? Absolutely not. We're going to get half blood type A, half blood type B, because A and B are dominant over blood type O. Right, so here is a question. If a boy has a blood type which is O and his sister has blood type AB, what do you think the genotypes and the phenotypes of their parents will be? Now what you should do is you should pause the presentation at this point and try and work it out. The first step though would be to write out the genotypes of the boy and his sister. And that's what it would look like. So you have type O, which we know the genotype of, and we have type AB, which we know the genotype of. Alright? So what do? We have to lay out our Punnett square to try and figure out what the genotypes and phenotypes of the parents will be. So here it is. We laid out AI, IB, that's what it would look like. And we know this is what the children will have. So the parents must have genotypes IA, IB, which means that the parents are, in terms of their phenotype,